Welcome back, everyone. We're going to continue on with Chapter 8, Molecular Biology. So in our last video, we started out with protein synthesis with transcription. So now that we understand how we take our DNA and we transcribe it onto RNA, particularly messenger RNA, we're going to take that messenger RNA out into the cytoplasm where it binds with ribosomes and reads the code from the mRNA to translate it into a protein. Okay, so the amino acid sequence. Okay, so that's what we're going to learn about in translation. So again, we're going to focus in on the second part of protein synthesis, which is translation. So this part is probably the cell's most energy consuming metabolic process. So this is where it's making proteins. And proteins, if you think about your body mass, proteins make up the largest body mass of any component. Okay, so proteins make up the most mass of you, with the exception of water. So, and proteins, as we learned about way back in what was it, chapter 3 or chapter 2? I think it was chapter 2. They perform a variety of functions within your body. I mean, there's structural support. They provide, there are enzymes within our body. They provide immune support. They are part of communication. They do all sorts of things within our body. So the process of translation or protein synthesis, we're taking that mRNA message and we're translating it into the polypeptide chain. So we're going to learn about that process. So by the end of this video, you should be able to describe the different steps in protein synthesis, as well as describe the role of those protein factories, the ribosomes, in protein synthesis. And then you should be able to describe the genetic code and how the nucleotide sequence determines the amino acid and the protein sequence. Okay. So we're going to start out this video with an Amoeba Sisters video. So this Amoeba Sisters video talks about protein synthesis. So it will start out with a review of transcription, and then it goes into translation. But this one, it covers translation very well. So it's a good video to start out with.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on the pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins, which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in, and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You can see on the codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So again, that was a great introduction into translation, as well as a good review of what we learned about transcription in the previous video. So now let's get into the details. So let's start off by talking about RNA. So we already know a little bit about RNA from the previous video, specifically messenger RNA. So we learned about messenger RNA as the RNA that is being created or synthesized from the DNA template inside the nucleus. Okay, so that's what transcription is all about. So it's that form of RNA that carries the nucleotide sequence code for the protein sequence that's translated into the polypeptide sequence. Okay, so here is basically represented here in this diagram from your textbook. Actually, I don't think this was from the textbook. Yeah, it is. Okay, from the textbook. And you can see each one of these little purple dots represents one of the nucleotides. So it could be the A or the G or the C or the U. Okay, so each one of these represents one of the nucleotides. Okay. Next, we have the rRNA. So that stands for ribosomal RNA. So ribosomal RNA makes up the ribosome. Okay, so the ribosome, and if you recall, wow, way back in, I think it was chapter 3 when we talked about the parts of the cell, we talked about the nucleus. And remember, inside the nucleus, that dark splotch called the nucleolus is where the rRNA is made. Okay, so the nucleolus is where the ribosomal RNA is made, and that is where ribosomes are made. Okay, so because it's made of rRNA. So ribosomal RNA originates from the nucleolus of the nucleus of the cell. Okay, and so it's made of rRNA. Okay, so this rRNA, you can see in this picture here, ribosomes are made of two parts. We have a small subunit and a large subunit. And you can see where the mRNA sits. It's right in between the large and the small subunit. Okay? And then finally we have the third type of RNA. We have transfer RNA, otherwise known as tRNA. So tRNA, if you look at it, it's supposed to be in the shape of a T. That's why it's called tRNA. But it also stands for transfer RNA because it transfers a specific amino acid to the growing polypeptide chain. And so there's a couple parts to this tRNA. So one part down here contains what's called the anticodon. So we have something called a codon. We'll talk about this later. But okay, so the codon is basically, it codes for a certain amino acid. And so the codon, if it codes for a certain amino acid, the anticodon is just the complementary base pair for the codon. So for example, if there's an A here on this mRNA strand, then on the corresponding tRNA, there will be a U. Okay, so the U binds with the A. If there's a G here, then a C will bind to it. If there is a C here, then a G will bind to it. So it forms like this complementary base pair. So the codon has an anticodon, that the anticodon kind of binds with the codon. Okay, so that transfer tRNA, it knows where to link onto on the mRNA strand, okay, to bring the correct amino acid to the ribosome. And so that tRNA, it's transferring in a specific amino acid to the ribosome based on which codon it can link to based on its anticodon it pairs with. All right. So those are our three types of RNA. So we have the messenger RNA, that's that sequence of RNA nucleotides made off of the DNA template. We have the ribosomal RNA that makes up the ribosome. And then we have the transfer RNA that basically transfers the correct amino acid to the ribosome based on what the mRNA code says to bring to the polypeptide chain. Okay. So that's part of what we need to get in our brains before we put everything together. So that's the machinery. Next, let's talk about this genetic code. So the Amoeba Sisters video talked about the code. So first off, in our proteins, we have 20 commonly occurring amino acids that can make up a protein, so a combination of these amino acids. And as we know, on RNA, there are 4 different possible nucleotides. We have adenine, guanine, cytosine, and uracil. So if we have 20 commonly occurring amino acids and we have 4 nucleotides, we have to figure out, how many possible, combinations can there be to make, enough so that we get 20, enough of the combinations of nucleotides in order to get the 20 commonly occurring amino acids covered. So we know, we can't do just 4 because, that's not going to cover it. We need 20. We need 20 combinations. If I go, if I just have 2 amino acids in a code, so just, if I just take 2 amino acids, if I do all the different possibilities of combining all of these, out of 4 types, all of these possibilities, I can only get 16 different combinations of possibilities. So that doesn't add quite up to 20. But if I do a combination of 3 nucleic acids in a row, I can get 64 possible combinations. So that'll give me enough combinations to get well above 20 possible combinations to get to the 20 that I need for my amino acids. So that's how the geneticists figured out the codon chart. So if we look at this codon chart, we come over here and we look at all the possible combinations of, 3 nucleotides out of the 4 possible, letters here. So there's all 64 possibilities. And they figured out that, if there's 3 U's, uracil, 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 it will code for a specific amino acid. Okay, if we put uracil, cytosine, and uracil, this codes for a different specific amino acid. CCU, cytosine, cytosine, uracil, that codes for a different specific amino acid. So each one of these triplets represents a codon. So the 3 nucleotides on the mRNA strand represent a codon. Okay? And you can see that each triplet codes for a specific amino acid. You'll never have a triplet code for multiple amino acids. You won't see UUU code for like 2 different amino acids. It only codes for one specific amino acid. However, you will notice that you'll see some amino acids have multiple codons coding for it, and that's because, you know, we have extra here, right? 64 possible combinations of our nucleotides, but there's only 20 possible amino acids. So we do have multiple amino acids, or one single amino acid coding that are coded for by multiple different codons. Okay. Another thing you'll notice from this codon chart that we do have one codon that is known as the start codon. It stands for methionine, like you saw from the video. And this is where the ribosome knows where to start, okay? Where to start reading the mRNA and where to, you know, start making the protein, okay? So that's the start codon. So that's where it starts reading it. And then, you know, it'll start reading, and then it might see, you know, this triplet, so it knows to put leucine there. It might see this one, it knows to put proline here. It might see this, and it knows to put lysine there. And you can see that these are just abbreviations. You don't need to know the full amino acid, okay? And then eventually, it's going to come across one of these 3 stop codons. So these stop codons on the mRNA strand, they don't actually code for an amino acid. Okay, so once the ribosome reaches one of these stop codons, there's nothing, no amino acid coded for. So that's when it releases the polypeptide, and then the whole polypeptide is done growing, and the whole thing's done. Protein synthesis is complete. Okay, so this is basically our genetic code. Now, this genetic code is universal, so it doesn't matter if we're looking at the RNA of a human or a bacterial cell or a banana or a, you know, plant or a pig or a duck or a dog or a cat or, you know, whatever. It's universal, okay? So it's all the same. So if I look at human DNA and I read CUG, a leucine amino acid will be placed there. If I look at a bacterial DNA and I see CUG, a leucine will be placed there. Okay, so it's universal. It just shows that we are all related. We all have a common origin there. Another thing that you'll want to note is that this genetic code, you never have to memorize anything from here, okay? You just know how, you need to know how to read it. So if I ask you to, you know, if I give you a sequence, like for example, let's say that I give you the sequence A, U, G, C, G, A, U, A, G, then I expect you to use this codon chart or the genetic code to be able to translate it. Okay, so can you do that? Well, let's see. AUG, AUG. And you can, if you really need it, the first letter is on the side. Okay, so that's the first letter A, second letter's up top, U, and then third letter is over here, G, so AUG. So that stands for met or start. And then the next one, CGA. What does that one code for? See if you can find it. Were you able to find it? So that one's arginine, ARG. And again, you just need to abbreviate. You don't need to actually spell out arginine. Okay, and then the last one, can you find that one? That one's one of the stop codons. So you don't even have to put anything there. It just stops because it doesn't code for anything. So how many, so this is our amino acid basically chain here. That's it. All right. So that's how we read our genetic code. Hey, we did some protein synthesis already. So we've learned about the machinery, the protein machinery. We've learned about the genetic codes. Now we're ready to put it all together. Okay, so let's start out with the mechanism for protein synthesis. So just like in DNA replication and in transcription, we have three main stages. We have initiation, we have elongation, and we have termination. So it's so easy to remember these things. They make it so simple. So let's start with initiation. Okay, so initiation, we have our mRNA template. So here's our mRNA template that we created way back in transcription out in the nucleus of the cell. So it's made its way out into the cytoplasm. And you can notice that, here's the 5' end. So hey, where's that 5' cap over there? And remember, there's a poly A tail over here on the 3' end. And then here is the sequence. We got AUG, UUU, G, CGA, blah, blah, blah. Okay. So the mRNA template is going to start to interact with the small ribosomal subunit. So remember, the ribosome has two parts to it. It has the small ribosomal subunit and the large ribosomal subunit. So it's going to start interacting with the small one first. So there it is. It's interacting. That's the small one down there. And so, hey, it's interacting with it. Next, we have a tRNA. So here is a tRNA. Doesn't look like a T for some reason, but oh well. So we have our tRNA, and this particular tRNA has the anticodon UAC. So what does UAC bind with? Well, remember the U binds with an A binds with a U, and a C binds with a G. Hey, that's our start codon, right? Okay, so the tRNA is going to bind with the start codon, and hey, it carries the methionine amino acid. So this tRNA, it's bringing over the methionine amino acid, so that's attached to the other end of the tRNA, and then the anticodon is going to, have a temporary bond with the codon of the mRNA. Okay? And that's it. That's the initiation stage. Okay. So next we're going to move into, what's the next stage? Elongation. Okay. So here's the first stage. Initiation's up there. So the next stage we have elongation. So that's when we have our small subunit. Well, the large one joins into the party. So here's the large subunit moving in. And this large subunit actually has docking stations in it. And these docking stations contain 3 sites to it. It has an A site, a P site, and an E site. Okay, so the A site is the incoming site. Okay, so this site's kind of the far right site. This site is for the incoming tRNA strands. Okay, or the tRNA molecules. So when a new tRNA molecule comes in, it's going to dock at the A site. Here, let's look down here. I like this view better. Okay, so here is our large ribosomal subunit. So the next tRNA is going to move in and dock at the A site. So here's our A site. And you can see how this particular tRNA has the anticodon GCU, and it's going to, have a temporary bind with the codon CGA. So that matches up, and hey, it's bringing along this amino acid there. Okay, so it's going to click into place. We have next to the A site, we have the P site. So the P site is where there's a tRNA already there. And that tRNA is attached to the growing polypeptide chain. So you can see, we already have that methionine, okay? So that methionine is there, and then we had like another one already come in. So we already have like a growing polypeptide chain forming here. So the middle one, the P site, is where the growing polypeptide chain is attached to, okay? And then the E site, think exiting site, okay? So that's where basically where the tRNA that doesn't have anything attached to it, okay, that its amino acid's been released, is leaving, okay, it's exiting, right? So I always think like the A is the arrival site, the P is the polypeptide chain site, and then the E is the exiting site. So that's how I remember the three. Okay, but this is basically the process. So as the ribosome is moving along, it's going to be reading the mRNA, okay, and then the next tRNA is going to come along and dock in the A site, okay, and bring that new the new amino acid into the ribosome. Okay. Meanwhile, at the P site, okay, the P site is going to take whatever was, previously at the A site, transfer that over onto the growing polypeptide chain. Okay, so that's growing along, and then whatever was here gets, moved into the E site where it leaves. Okay, so everything is going to just keep shifting as the ribosome moves along. So then whatever was in the A site shifts to the P site shifts to the E site. Anything here gets shifted to the P, any polypeptide, growing polypeptide stays at the P site. So we keep moving, the polypeptide chain is forming along the way. All right. And then finally, once we reach a stop codon on the mRNA strand, remember one of those stop codons, UAA, UAG, or UGA, those don't code for anything, okay? They don't code for an amino acid. So when that gets reached, then the growing polypeptide is released, okay? Because basically, there's nothing that codes for it, so it just ends. And so basically when that nothing will be in the A site, and since nothing's in the A site, then that tells the P site that, we're done here, and this will get detached. And then now we have our polypeptide is released. And then the tRNAs will, go back and do their thing. The mRNA is degraded and the nucleotides get reused. And then the ribosomal subunits disassociate. Okay? So that's when everything's terminated. Okay? So that's pretty simple. Okay, so let's do some review questions now that we understand our translation. So the RNA components of ribosomes are synthesized in the, so where are ribosomes synthesized? Is it the cytoplasm, A, B, the nucleus, C, the nucleolus, or D, the endoplasmic reticulum? So where are ribosomes made? Where's the rRNA made? So if you remember way back in chapter 3, the nucleolus is where the rRNA is made. The nucleolus, that dark splotch in the nucleus. All right, here's another question. How long would the peptide be that is translated from this mRNA sequence? Okay, so take a guess of how long. So if you said 4, I would agree with you. So how did we come up with 4? Well, remember The codons are triplets. So here is the first codon, which happens to be a start codon. Here's the second codon. Here's the third codon, and then there's the fourth. So each one of these codes for an amino acid. So there's 4 peptides or amino acids in this sequence. So 4 is the correct answer. All right, so we are done with translation and protein synthesis. So by now you should be able to describe the different steps in protein synthesis. You should also be able to describe the role of ribosomes in protein synthesis, as well as describe the genetic code and how the nucleotide sequence determines the amino acid and protein sequence. Okay. So to complete chapter 8, we're going to, in the next video, learn about how genes are regulated.